Okay, to begin where we left off, I was talking about the three shapes of bacteria, which are Spirillum, Bacillus, and Cassis. The Spirillum are twisted or spiraled. The Bacillus are rod-shaped, and the Cassis are spherical shaped. So here we see Spirillum bacteria at 1,000 times magnification. Even at 1,000 times, it's hard to see, but you can tell that the bacteria are wavy, which indicates that they are twisted and stringy and so forth. Here we see a bacillus. As you can see, they're much straighter. They're thicker, which you can see they're shaped more like a rod, which is what bacillus bacteria are. Here we see Cassis bacteria at 400 times magnification. Even though it's not very clear to see, you can tell these bacteria are more spherical or round shaped, and I believe if we magnified it more, excuse me, that you would be able to make out the spherical shape better. We'll see a better example of a spherical shaped bacteria shortly. This is Staphylococcus at 1,000 times, and again, it's very hard to make. It's fairly hard to make out really what the shape is, but here on this slide we can see that it's very round, and this is only 40 times. This is live Staphylococcus, and you can tell that it's Cassis bacteria. It's in the name. So, and it's round, so that would indicate that kind of bacteria. The next kingdom is Protista. They are eukaryotes, which are almost always found in water. If it's water-based, wherever there's water, they have to be in water. They're also organotrophs. They can reproduce either asexually or sexually. And they impact humans in a couple ways. One example is malaria. Plasmodium is a protist that causes malaria. We'll see that very shortly. And some protists can cause sleeping sickness, which is a parasitic disease caused by a protist. Here we see a blood smear of Plasmodium. A blood smear infected with Plasmodium at 400 times magnification. And next we had to prepare a wet mount because the next step was to obtain some pond water, observe it, and find a protist living inside of it. Pond water would be pretty useful. So what we had to do to prepare this was get a slide, add a drop or two of pond water, and place the cover slip over it. That's how we made the wet mount for pond water. Here you can see a video of protocells moving, and this was at 40 times magnification. There's one right here. I believe there's also one right here. I'll replay that because my computer is awfully slow and you might not have been able to see that. This little dot right here is moving, and so is this one. Alright. So the final kingdom was Archaea. They're prokaryotes, which are generally found in extreme conditions: very cold, very hot, very salty, very saline. Yeah, they reproduce asexually. An example, binary fission. They are another example of chemoautotrophs. An example of Archaea would be halobacteria. They live in saline environments, very salty, and they impact humans. They are found in the human colon, in which they act as methanogens and they aid digestion. And methanogens, what they do is they release methane gas, which we know has a very foul odor, and I think we know where that ends up. So our conclusion to this is we looked at red blood cells. These are also known as erythrocytes. They are eukaryotic cells which carry oxygen and carbon dioxide throughout the body. And we use microscopes to view red blood cells at 3 different magnifications. And as the field of view decreases, our magnification was increasing. As we increased the magnification, you saw less of the slide, but you could see it better, which is you can observe the individual cells at 400 times. You can almost see the donut shape, which I have a picture in the next slide, and we know that erythrocytes are anucleated, which is uncharacteristic of most animal cells. Here you can very vaguely, I don't know if it's clear to make out, but see our dark patch in the middle, because we know that red blood cells in this state have their nucleus removed, so they have a well in the middle. So the cheek cells are, of course, eukaryotic. They're from Animalia, a human, me. They have a nucleus. As you can see, this is 400 times the magnification. This dark blue dot is the nucleus. We definitely know this is a eukaryotic cell because it's a very compact region. And also notice that this cell does not have a cell wall. But, moving on, Elodea cells, which are also plant cells, do have a cell wall, which is one defining characteristic of plant cells that differentiates them from animal cells. So the Elodea cells, they're eukaryotic. They're from the kingdom Plantae. Using microscopes, we can see chloroplasts, the tiny dots along the cell wall. Another feature that distinguishes plant cells are the chloroplasts, which are the photosynthetic organelles. You can see them along the cell wall here. So there were also protist cells that are eukaryotic. Of course, we know they fall under the kingdom Protista. They're so protists could be unicellular or multicellular. Using the microscope, we can magnify the pond water to see movement of a protist. And we also observe Plasmodium, which causes malaria in humans. So next we looked at fungi, which are eukaryotic under the kingdom Fungus. We have observed Saccharomyces cerevisiae, which is yeast, Rhizopus, and Aspergillus. The slides we were given for the fungi, except for the yeast, had they could either be Rhizopus, Aspergillus, or Penicillium. And doing a little research and finding images that compare to the pictures we took, we determined that we looked at Rhizopus and Aspergillus based on the structures we saw. So next there was bacteria. They're prokaryotic, fall under the kingdom Eubacteria. We observed the 3 common shapes of bacteria, Spirillum, Cassis, and Bacillus. And we also observed Staphylococcus aureus and live Staphylococcus aureus. And we also concluded that Staphylococcus aureus is a Cassis bacteria because of its round shape. And archaea, well, we didn't observe any prokaryotic archaeal samples, but we know that they live in extreme conditions and they do play a role in human life. So that is the end of our presentation. And let's see. I have a football game and a vice presidential debate to watch, so I'll be going, and hopefully Michael Vick doesn't fumble this weekend.